Okay, you are ready to go.
Thank you.
As soon as you're ready to roll, turn that on, and you'll be good. Okay? And you see it's recording.
Okay, so I've already put on.
I've already done that. Perfect.
Thank you very much.
Okay. I'm bailing out on you.
Okay. Good luck. Grab some food on the way. Grab a sweet or two on the way out. Oh, okay. It's our secret. Hi, Susie. How you doing? Good. Thanks for coming.
It's a real pleasure and honor to introduce my MD PhD graduate student, Paul Parcon, to you all for the defense of his thesis. Paul and I have just had a really wonderful relationship because in many ways I have never admitted this to him before, but we're a lot alike. And one of the ways that we're a lot alike is we do have to confess that we do that collusion thing. So as the talk proceeds here, which is on aging and neuroinflammation and the effects of the inheritance of the principal gene for risk for Alzheimer's disease, the topic for the presentation today. And wonderfully, we had people interested from different groups, and our own group in particular, in the project. And so there was a lot of colluding, and it was all great fun, and just what science ought to be like. So it's my pleasure now, Paul, to come.
Thank you, Dr. Griffin. Thank you, everyone, for coming to see here. My name is Paul Parcon, as you heard. This is our presentation today about this thesis. We've titled it Neuroinflammation, Age, and ApoE Epsilon 4 Affect Autophagy in Alzheimer's Disease. And I'll kind of give you an overview of all the individual topics here and go into the data. So this is the general structure of our talk. First of all, I'll talk about Alzheimer's disease. And then I'll talk about autophagy and what it is. And basically, it's a biochemical process by which things like protein aggregates and unused organelles and things like that are digested and removed from the cell. Then we'll go into some data about protein aggregation in the AD brain, and then looking at neuroinflammation, especially IL-1, aging and transport and retrograde transport of these things, TFAB and the transcriptional regulation of Alzheimer's disease, and the conclusions and future directions. So first of all, what is Alzheimer's disease? AD, or Alzheimer's disease, I'll be calling it AD throughout the talk. It's the most prevalent neurodegenerative disease. It's the most common form of dementia. Now it's actually also the 6th highest cause of death in the United States. It's associated with a progressive dementia, and it's characterized by plaques that are outside the cell, containing amyloid beta, mainly amyloid beta, and neurofibrillary tangles, also inside the neurons, and that are made of this protein called hyperphosphorylated tau. Risk factors for Alzheimer's disease includes age, female gender, genetics, neuroinflammation, and especially this gene called ApoE epsilon 4. So I just wanted to show a few pictures here of the plaques and tangles that are in Alzheimer's disease. This is some of the staining that we've done here in our lab. In the red on the left-hand image is oligomeric A beta, which means that there's 6 to 10 amyloid beta molecules. And we've stained them here to show 3 different plaques in the Alzheimer brain. On the right-hand image in the red is the hyperphosphorylated tau, or these neurofibrillary tangles. And you can see these processes surrounding this plaque-like area right here. And you can even see in this corner here a flame neuron, which is a neuron that's full of hyperphosphorylated tau neurofibrillary tangles. So these are the characteristic protein aggregates that are associated with Alzheimer's disease. You have to have these in order to be diagnosed post-mortem. So this leads us to our hypothesis then, that factors that increase the risk for Alzheimer's disease impact the autophagy system, leading to the accrual of protein aggregates in the neurons and outside the neurons as well. And sort of the corollary for this is that interference with autophagy at any step, since it's a disease characterized by protein aggregates, could increase the risk for Alzheimer's disease. So we'll kind of go through the steps of autophagy, and we'll also go through how they correlate to the risk factors for Alzheimer's disease that we mentioned. So to just go into the weeds a little bit about how autophagy proceeds. So I mentioned that it's a degradation process that gets rid of misfolded proteins or protein aggregates and that kind of thing. So here in the green, you can see depicted misfolded proteins. And they can form into protein aggregates, as shown right here. The first step in this is going to be ubiquitination, which is a protein tag that the cell uses to tell itself to get rid of these protein aggregates. And these ubiquitins are shown here in purple. So a complex machinery of enzymes will attach these protein ubiquitins onto the outside of these protein aggregates, including an E1, E2, and E3, especially one called Parcon, which we'll talk about in a little bit in detail in the future. After the protein aggregate has been ubiquitinated by all of these purple ubiquitins here, we'll attach these little goldfish p62 bodies, which can bind to ubiquitin and themselves. And once enough of these p62 bodies surrounds this protein aggregate right here, we have the formation of something called a sequestosome. It's also another name for p62. Then p62 can also bind to this membrane-bound protein called LC3, and that's depicted here in these gray dots. And LC3, again, is membrane-bound. It's associated with lipids, and it can wrap this protein aggregate in a membrane, which is formed, which is called an autophagosome when it's fully formed, right here. And once this autophagosome has been formed, again, with the protein aggregate in the middle, surrounded by ubiquitin, and then p62 and LC3, it can be transported to the lysosome, which is this depicted here. And this is basically an acid-filled sac with proteases and things that can degrade proteins. And these lysosomes are tagged with a protein called LAMP2, which will become important later. So once again, we have the ubiquitination, sequestosome formation, autophagosome formation, and then fusion with the lysosome in order to degrade these proteins. So I mentioned all of these steps, but we're talking about autophagy, and it occurs in every cell. But neurons in particular have sort of an elongated morphology. So you could see here at the synaptic area right here, we have a very, very long axon, which could be a few feet sometimes. And we also have the cell body where the nucleus is and the DNA is located. And in Alzheimer's disease, we have basically issues in every step of this. And if we look at autophagy dysfunction, if we compartmentalize the cell into these three parts, here at the synaptic level, let's say, for the sake of this paper, that we'll have the protein aggregation occurring here, ubiquitination will occur here. And once the autophagosome is formed, it must be transported in a retrograde fashion along the axon on these microtubule tracks, and then it will be fused with the lysosomes here towards the cell body. Another thing that happens that's important here is the transcriptional regulation, which happens inside the nucleus of this neuron. So talking about protein aggregation, again, for our purposes, will be at the synaptic level. We wanted to look at that hyperphosphorylated tau protein that we were talking about previously. And in this slide, we employed a technique that I'll be going to several times throughout this talk. It's called PLA, or proximity ligation assay. And the proximity ligation assay will produce a colored dot when two proteins are within 40 nanometers or 400 angstroms of each other, so when they're very close. If they're too far apart, then they will not produce this dot. So in the first row here, we see that phosphorylated tau and sequestosome 1, which is that fish protein I saw earlier, are co-localized with each other in Alzheimer's disease brain. So you can see these right here. And if we tag sequestosome 1 in and of itself, we also show these. And this is showing that tau is being a substrate for autophagy, or it's being aggregated with sequestosome 1. In the second row right here, we are seeing hyperphosphorylated tau along with amyloid beta, so which is that protein that's in the plaques. And so it seems that p-tau and A-beta are co-localizing with each other. And in this set of slides, the green is just to depict that these are actually occurring inside of a neuron. I'm trying to show that this is happening inside the cell. So we know now that tau and amyloid beta aggregate with each other, and they're associated with the sequestosome 1 protein. We also wanted to look at autophagosomes themselves, and we can do that by tagging LC3, which is that membrane-bound protein I mentioned a second ago. So you can see here in AD with the PLA, this is hyperphosphorylated tau and oligomeric A-beta co-localized in these dots right here. If we overlay a stain for autophagosomes, you can see that these dots become yellow, which implies that these are in or around autophagosomes. Compared to an AMC and age-matched control, which is a patient that does not have dementia of similar age, we don't see very many of these dots at all. If we were to zoom in on one neuron, for example, you could see here in E the PLA dots that show p-tau and oligomeric A-beta on their own. And if we were to overlay this with autophagosomes in green, you could see that some but not all these autophagosomes contain an aggregated p-tau and oligomeric A-beta. Another thing we were interested in is looking at Parcon, which is that one of those E3 ubiquitin ligases. Now, its job is to attach ubiquitin to these protein aggregates or organelles that we need to get rid of. So in A, the first image here is we're showing a staining of Parcon, and it seems to be fairly regularly distributed throughout the neurons here. In B, we're seeing that Parcon has been aggregated in Alzheimer's disease. So it's forming these sort of rosette things. Then we did a PLA of Parcon with hyperphosphorylated tau, that tau protein again. And in the age-matched control, that is a patient without dementia, you can see that there's very little signal, very little brown here. Whereas in the Alzheimer's disease brain, we see Parcon is aggregated with hyperphosphorylated tau to a good degree, and this neuron, for example, is really filled up with it. So all of this suggests that p-tau and oligomeric A-beta could possibly be targeted to autophagosomes. They could be aggregated with each other. And another aspect that we were interested in is how this is affecting in a mouse model. So then we moved to a mouse model of AD. It's called the PDAPP mouse model. It's one of the earliest models for Alzheimer's disease. And we wanted to look at the mRNA level, or the transcript level of this protein. So we see here that compared to their non-transgenic littermates in the wild type, the PDAPP mouse model has elevations of Parcon mRNA both at the 2-month age and the 17-month age, so even at the young age. The amyloid plaques in this model actually don't appear until, I believe, the 4th month. But in addition to Parcon being elevated at the mRNA level, also this other proteins, neuroinflammatory cytokine IL-1 alpha and IL-1 beta, are also elevated alongside. So this led us to the question of how does IL-1 and Parcon associate with each other? Is there some kind of relationship? So we looked at these cells here that in the human brain, again, with Alzheimer's disease. And this time Parcon is depicted in the blue. And you can see once again that we're getting these very dark protein aggregates of Parcon. And in the brown is IL-1. So it seems like these neurons that are expressing aggregated Parcon are also surrounded by these glia that are expressing IL-1. So we took a cell culture model of NT2 cells, and we treated them with IL-1 overnight. And you could see here that the Parcon is at some baseline level in the untreated cells, which is quantified here. And treatment with IL-1 raises the amount of Parcon. So it appears that IL-1 is upregulating Parcon activity by increasing Parcon levels. And another way of activating Parcon is by this process called netylation, in which Parcon is attached to NED8, which is a ubiquitin-like protein, and this has been shown to increase activity. Now, this is from another person's work whose citation disappeared. I apologize. You could see that Parcon is expressed in these lanes here. Synphylin, which is a Parcon substrate, is expressed here. And then we have varying levels of NED8 here. So in this first set, we are showing that synphylin is being ubiquitinated to a greater degree the more NED8 that you have. And then this shows here, this is some level of NED8, this is a little bit more NED8, and again, you're seeing more of this activated, or this ubiquitinated synphylin, which implies that netylation of Parcon increases Parcon activity. So we wanted to go into some of our computational modeling, which is done by my good friend Sundram over there in the middle. Hey, Sundram. We took Parcon, which is depicted here in green and in red, and we highlighted in red the active site region ring 1, which is necessary to associate with the E2 ligase, E2 proteins in ubiquitination. So we wanted to see what happened without NED8. So we took Parcon, and then we put the E2 enzyme called UBCH8 here, and it appears that Parcon and UBCH8 localize in this fashion, where there's a distance between UBCH8 and the Parcon ring 1 domain of about 6 to 14 angstroms. And we're classifying this as likely inactive, or we predict that it's likely going to be inactive because of the distance is too far for a molecular bond. But now let's go with Parcon and NED8. So here's Parcon again depicted, and we put NED8 here, and you can see that it localizes to this area of Parcon. And when we zoom in on this, we can see that lysine 76 residue, this residue right here, is the one that is interacting most closely with NED8. And I didn't put the slide up, but there's also been some mass spec studies that showed netylated Parcon protein would be expected to bind to lysine 76. So in other words, this model is accurate. And then when we try to combine this with UBCH8, this time UBCH8 will localize to this aspect of Parcon, and the distance between these two is less than 3 angstroms. So we're saying that this is probably predictive of a more active Parcon complex. So in other words, NED8 is activating Parcon by its conjugation, at least by binding to lysine 76 right here. And this is just the whole model one more time. So here's Parcon on its own. Without NED8, you have UBCH8 on this side, 6 to 14 angstroms away. Here, with NED8, we see that it's less than 3 angstroms. So we're saying this is probably a more active Parcon complex. So what does NET8 do in Alzheimer's brain? So we just decided to stain it. And here you see a non-demented patient, age 76 years old, and we had stained NET8 with green, so you can see it right here, and it's localized to the nucleus of these proteins. So this is a non-demented person. Again, NET8 is in the nucleus. But if we look at the patient with AD of similar age, this person is 74, you could see that the NET8 is no longer in the nucleus. These nuclei, in fact, are somewhat empty, and there's a lot more of the NET8 that's in the cytoplasm. And we quantified this over several cases, and we found that there is a significant reduction in the amount of nuclear NET8 in AD compared to non-demented control patients. So this is kind of curious. Why is NET8 localized to the cytoplasm instead of the nucleus in AD? So we thought, okay, maybe Parcon is more likely to be NET-lated. So here in this first image, a 71-year-old control patient again, we did the PLA study that shows that NET8 and Parcon would be co-localized to give us this red dot right here. And you can see that there's some level of that in the age-matched control patient. But if we look at the AD patient, we just have a lot more of this NET-lated Parcon. They're very close together. And again, we quantified these over several cases, and we see that there is indeed more NET-lated Parcon in Alzheimer's disease brain compared to age-matched control brain. So that's curious. We wanted to see what's going on in Parcon, and we know that IL-1 is upregulated alongside Parcon. So what is IL-1 doing to NET8? So then we took a cell culture model here. These are primary neurons, and here you see that the NET8 is also in the nucleus. They're having this really nice teal color right here in untreated cells. But if we treat these cells overnight with IL-1 beta, we have this co-localization here where the NET8 is still in the nucleus to some degree, but a lot more of it is moved out to the cytoplasm, and the nuclei appear to be less dense. So we wanted to quantify this in a couple of different ways since we have the cell culture model. And using immunohistochemistry or immunocytochemistry with the same way that we did it with the AD brain, you could see here that in the untreated cells we have some level of nuclear NET8, and with IL-1 beta treatment we have a reduction. Here, the cytoplasmic NET8 is at some level again, and then with IL-1 treatment, we have an increase. This is consistent with the idea that IL-1 is leading to a nuclear to cytoplasmic translocation of NET8. So that's by the immunohistochemistry model. We also did it by subcellular fractionation. So we separated the nuclear protein from the cytoplasmic protein here. And on this Western blot, you can see here the cytoplasmic NET8 is at some level, and that's shown to be cytoplasmic by the actin, which is a cytoplasmic protein. And here is the nuclear portion of NET8, which there is some existing. There's no actin in the nucleus, but there is histone 1. So this is kind of a proof of principle that this fractionation worked. And in the untreated cells, you could see that there is some level of nuclear NET8, again, that has been decreased by treatment with IL-1. And there is some level of cytoplasmic NET8 that is increased by treatment with IL-1. So again, all of this data is consistent with IL-1 leading to a nuclear to cytoplasmic translocation of NET8. But to what purpose, right? So we see that NET8 is associated with Parcon. So we wanted to look at these cells now. This is a different cell culture model called the NT2 cell model. And we did the PLA dots one more time to see NET8 and Parcon in the untreated cells. And in the treated cells, we have a lot more of this signal in the NET8 of IL-1 treated cells. In other words, not only does IL-1 increase translocation of NET8, it increases Parcon netylation as well. And it's very clear in these pictures, but we wanted to quantify it as well, and we see that there's more Parcon netylation in the IL-1 beta treated cells. So that's all very interesting. IL-1 seems to activate Parcon for increased ubiquitination and that kind of thing. But I wanted to put in this other aspect of it. This protein kinase called PINK1 phosphorylates Parcon. It leads to its open configuration, and it also phosphorylates ubiquitin, which is an activator of Parcon as well. So this is what PINK1 does. And if you're wondering what PINK1 fits into our model here, so this is our model once again. PINK1 actually starts right here because this model took into account that serine 65 on Parcon of this version of Parcon is already phosphorylated. So PINK1 should go here at the very beginning. So what does IL-1 do to PINK1, which is going to be very interesting, I'm sure. IL-1 beta actually reduces PINK1, which is kind of strange. So we see here that in primary rat neurons and in NT2 cells, the amount of PINK1 at the full length and of the cleaved portion, it's decreased compared to their untreated counterparts. And in NT2 cells, we weren't able to detect the cleaved portion, the 52 kD, but the full length portion is reduced in IL-1 beta treatment. Further, how does NET8 and PINK1 associate with each other? So we once again did some modeling. On the left-hand side, you can see PINK1 depicted in green, and its substrate ubiquitin inside this pocket, or this active site. And again, PINK1 will phosphorylate ubiquitin, which would activate Parcon. So here in the green on this side, we put NET8 in there, and NET8, being a ubiquitin-like protein, seems to fit into this pocket as well, but NET8 cannot be phosphorylated. So this is consistent with the idea that NET8 could act as a competitive inhibitor for PINK1-mediated phosphorylation of ubiquitin. So to summarize the effects of IL-1 on this system, it appears that IL-1 treatment increases Parcon level, its mRNA is increased alongside Parcon in the AD mouse model, and we have increased netylation, which would tend to increase Parcon activity. However, in PINK1, it's decreasing the PINK1 level, and it's also increasing NET8, which would inhibit the PINK1 active site. So this could be a reason why that IL-1-mediated neuroinflammation could be thwarting the autophagy that it seems to be trying to upregulate at the same time. And we'd actually done some work in some older work that showed that IL-1 is responsible for the upregulation of APP, which is the precursor for amyloid beta, and activates kinases to phosphorylate p-tau. So not only is IL-1 beta necessary for the production of the seed proteins of these characteristic aggregates, it also seems to thwart the clearance of these aggregates through autophagy. So to move on to the transport, I mentioned that ubiquitination will happen at the synapse level about right here, and then in autophagy, these autophagosomes then need to move back towards the soma right here. So we wanted to characterize this retrograde process, and this process is carried out by a protein called dynein, or dynactin P50. These are motor proteins that carry substrates back towards the center of the cell body right here. And we were interested in the effect that age, one of the other risk factors for Alzheimer's disease, could affect this process. So it's been shown by other groups that retrograde transport is less efficient as as an organism gets older. So we looked at our human population here, and we wanted to measure the amount of dynactin P50 here, which is depicted in red. And you could see that as the patients aged from 16 to 80 years old, the dynactin P50 levels also increased, which is consistent with the idea that this is sort of a compensation in age for the decreased dynactin efficiency. But if you look at dynactin P50 in Alzheimer's disease, again, this person here is 72, this person here is 69. The age-matched control of the non-demented patient has more dynactin P50 compared to its counterparts that have AD, which is consistent with the idea that Alzheimer's disease is failing to compensate for this retrograde transport. In addition, we wanted to look at how dynein could co-localize with other proteins. So we had APP here and NFTs in blue, and this combination of dynactin P50, APP, and p-tau could be shown by these different colored aggregates. And you can see that even though Alzheimer's disease had less dynactin to begin with, also the amount that's there is also going to be carried in these aggregates, no longer functional, one could say. So here in the dynactin P50 is another PLA experiment. It's depicted here in the pink, and you can see that dynactin P50 is in fact aggregated with p-tau. So one of the other risk factors for Alzheimer's disease is inheritance of the ApoE allele. ApoE is a gene that comes in three parts, E2, E3, and E4. And ApoE gene will code for a protein called ApoE, which is a lipid-binding protein, and those proteins are called E2, E3, and E4, and it's the greatest genetic risk factor for Alzheimer's disease. So we wanted to know how ApoE could affect autophagy in this system. So one of the first things we did was do the same thing with dynactin, and we stained dynactin here in a patient that has AD and has 3,3, so we're calling this AD33. Dynactin here is depicted in green, and you can see that even though they have Alzheimer's disease, depicted in green is the decrease in dynactin P50 in the AD44. At the same time of reduced retrograde transport, or reduced dynactin P50 at least, we see that they have an increased amount of hyperphosphorylated tau as well. So less transport back to the cell and more hyperphosphorylated tau. We did some Western blot studies on these as well, and you can see in these first 5 patients here, the AD33s, they have some level of dynein and dynactin and some level of p-tau, and the AD44s have less dynactin overall and more p-tau overall. So in summary, at the transport section, age seems to reduce the efficiency of retrograde transport, But in patients without AD, dynein and dynactin appear to increase to compensate, and AD brain has less dynactin compared to AMC brain. Dynactin was found to be aggregated in AD, and AD44 patients had more p-tau and less dynein dynactin. So this led us to the question of how exactly does ApoE lead to this issue? And it's sort of an interesting story. Dr. Griffin and I were talking about this, and I was looking online on some literature that showed that ApoE could also be associated with DNA to be a transcription factor. And the proteins that I mentioned here in the original autophagy overview, P62, LC3, and LAMP2, all of these are promoted by this DNA site called the clear site, which is the coordinated lysosomal expression and regulation site. And I happen to find that the DNA motif, that the clear site, is associated or very, very close to the one that is expected to be bound to ApoE. So we decided to check whether this happened. So let's go into the weeds a little bit about how TFEB, this transcription factor that's associated with upregulating autophagy, is regulated. So TFEB here, depicted in the yellow, is normally phosphorylated by mTOR1, and this phosphorylation binds it to 1433, and nothing's wrong with this cell. It's normal and it's fed. TFEB is phosphorylated and bound to 1433. Under conditions of stress or starvation, calcineurin will take the phosphate off of TFEB, which will allow it to dissociate with 1433 and enter into the nucleus, where it will bind with DNA, and it will upregulate the mRNA of the three proteins that we mentioned, LC3, P62, and LAMP2. So this is the whole model altogether. So we wanted to know what happens to these TFEB-regulated mRNA transcripts in Alzheimer's disease. So we looked at LAMP2 and P62 and LC3B, and we see that there is no difference between the amount of mRNA in the AMC versus the AD in any of these three. But since we know we were interested in ApoE, and we saw a difference between people with AD that had 3.3 or 4.4, we wanted to separate these Alzheimer's disease cases. And so we see here that AD33, the patients that have 2 copies of the 3 gene, actually have elevated mRNAs of all 3 of these, whereas their AD44 counterparts had decreased amount, and sometimes it might have been even less than the AMC. So this implies that the AD33s attempt to compensate for this, but the AD44s are not able to. So what's happening at the protein level? Again, this is the mRNA level here. The protein level, again, this is the mRNA on this slide. This is the protein. So AD33s have elevated mRNA and AD44s have less. And the protein level, they have more compared to their AD44 counterparts. So it appears that the defect in the AD44s occurs before the translation step. So here is depicted LC3B is another one of those proteins, and the AMC up here has some baseline level. The AD33 has an elevated level of these LC3Bs, and the AD44 actually seems to have less. But at the same time they have less, they also have more oligomeric A beta. So you can see here, this one does not have very much. It doesn't have Alzheimer's disease. The AD33 has more A beta and also more LC3. The AD44 has even more A beta but less LC3. So again, the protein seems to match the transcript level. So what is the TFEB localization? Again, TFEB needs to be in the nucleus to attach to DNA to upregulate these proteins. So we measured the amount of TFEB that's in the nucleus. And you can see here in the age-matched control patient, since it's not stressed, does not have Alzheimer's disease, there's some level of TFEB in the nucleus. But in both cases, the AD33 and the AD44, they both have an upregulated amount of TFEB in their nucleus. So it seems like there's some kind of response attempting to happen. Similarly, we looked at the cytoplasmic TFEB, which is 1433, and TFEB, again, can't go into the nucleus in that conformation. AMC33, not stressed, no Alzheimer's disease. They have a lot of these cytoplasmic complexes, but in the AD33 and the AD44, they seem to have less, which implies that the TFEB translocation machinery into the nucleus is working okay. So that implies to us that there was something going on between the step of nuclear translocation and transcription of these processes. So we did a cell culture model, and these cells were transfected by Dr. Barker. Thank you very much. And we have them expressing ApoE3 or ApoE4. So here in A, you see in this lane is insoluble protein aggregates. So that was done by my friend Dr. Ayadavaro. Hey, Srini. And we ran these on the gel and quantified the amount of protein aggregates in each of these cells. So in the second lane, E3, they have some level of protein aggregates in them. And E4, just at baseline, already has more protein aggregates. So even when nothing's wrong, theoretically nothing's wrong, these cells that express ApoE4 already have more protein aggregates. One way to activate TFAB is starvation. So this third lane here is where we starve the E3 cells, and like we expected, TFAB is upregulated, autophagy is upregulated, and the amount of insoluble aggregates is decreased in those cells that are expressing ApoE3. But under starvation conditions, those in ApoE4 don't seem to respond. That is, their aggregate amount does not seem to change. So we wanted to make sure that this is actually autophagy as opposed to a proteasomal degradation or something else. So we did some proteomics on the protein aggregates that are in these cells. And mitochondrial proteins in particular are only degraded by autophagy for the most part. And so these these mitochondrial proteins are in the E3s, there's some level, once again. When you starve the E3s, they decrease. When you have the E4s, they have upregulated, or there's a greater amount of them. And then with the starvation, the E4s actually don't seem to respond. They actually go up. So what is the mRNA doing? So it seems like there's no response in the ApoE treated cells. So we decided to measure those 3 mRNAs one more time in these cells that are expressing E3 or E4. So here are the normal E3 cells. They're sitting around, they're completely fed. When you stress them, TFAB seems to upregulate these 3 transcripts, as you see, which is what the expected problem is supposed to be. E4 at the baseline has some level, almost the same as ApoE4, the ApoE3 cells, but if you starve them, they barely seem to respond. So in other words, the cells that are expressing ApoE4 don't seem to be able to mount the mRNA response that we'd expect. So this, since we know that it happens after translocation to the nucleus, and we know that it's before transcription, then what's the only thing that could happen between there is DNA binding. So this led us to the question, does ApoE4 bind the clear sequence DNA? So Dr. Griffin and I were talking about this, like how could we possibly test something like this? And then my friend Sundram, who's a postdoc in Dr. Reese's lab, said, oh, well, if that's the case, if that's true, then I can model it for you. So he's, being a computational biologist, he did this model for me. Depicted here in the blue pipes is ApoE3. There we go. So here's ApoE3 in the pipes, and you can see that it's, has some movement, and DNA is depicted here at the bottom. And you can see that the DNA and ApoE3 seem to separate themselves from each other. And this is going to be a 50 nanosecond simulation, and ApoE3 and the DNA don't seem to associate at all. Let's compare this to ApoE4 here. And ApoE4 in the blue pipes, once again, is binding to the DNA very closely. And one thing I'd like to point out is if you look at one of the strands of the DNA right there, you can see that it is actually very stabilized. It's not moving very much at all. So this implies that ApoE4 is binding to DNA. So we wanted to characterize this model a little bit. So over the 50 nanosecond simulation, you can see that ApoE3 here, the distance between ApoE3 and DNA starts low, but will tend to increase and stay steady far away from ApoE and from the clear sequence in the black. And in the red lines, you can see that ApoE4 actually has decreased, gotten closer to the clear, and has stayed low for the rest of the simulation. The number of H bonds in ApoE4 is also more, the hydrogen bonds. There's more interacting residues in ApoE4 and the DNA, shown right here. And if you do the interaction energy, and less energy is a greater interaction, you can see that ApoE2 and ApoE3 have negligible amounts of energy, but ApoE4 has a lot less. So in other words, this is a much more likely conformation. We wanted to check whether ApoE4 and ApoE3 are binding just any random sequence, or if it has to be the clear sequence. So we took the clear sequence DNA right here and we changed the residues. In ApoE3, you can see that ApoE3 and the DNA have, again, no interaction with each other. ApoE4 and the mutated sequence also have no interaction with each other. So it implies that this is specific to the clear site. And we also wanted to change some residue, so we took ApoE4 and we changed this residue, and these two are the ones that seem to be binding to the DNA in particular. So ApoE4 depends on the arginine 112 and the arginine 61, because when you take them both off, they disappear, whereas 255 doesn't seem to work. So we solved it. ApoE4 binds to DNA, and that's what's happening, right? So there's a few questions that people have asked. Is there even ApoE in the nucleus? We know that ApoE is a protein that's normally secreted. It's a lipid-binding protein. So my friend Dr. Chanda Bose did subcellular fractionation on these. We see that ApoE3 cells and ApoE4 cells have some ApoE in their nucleus, and the cytoplasm as well. So lamin B is a nuclear protein, and you can see that ApoE3 and ApoE4 cells both express ApoE in the nucleus. So does ApoE4 bind clear in vitro? So Dr. Yadavara had these clear probes that have the clear DNA sequence and mixed them with proteins from the ApoE3 cells and the ApoE4 cells. And we see that the ApoE3 cells don't really bind the clear probes very much. There's not very much ApoE bound to clear in the 3 cells. But in the ApoE4 cells, there is a lot of ApoE bound to the clear DNA probes. So, okay, that's all well and good. We can bind DNA, but does it compete with TFEB for the binding sites? So we see here in the same assay, in E3, we have some amount of ApoE that's bound to this clear probes, and we have a bunch of TFEB bound to there as well. And the ApoE4 cells, however, don't have as much TFEB binding to clear. So it seems like that ApoE is binding to DNA, it's in the nucleus, it binds to DNA, and it competes with TFEB for the clear sites. So overall, our model is this. In TFEB is entering the nucleus here. If you have ApoE3 ignores DNA, but jumps off, and TFEB can attach to DNA and upregulate LC3B, P62, and LAMP2 to mount a normal autophagy response. Whereas in ApoE4 will bind the DNA, TFEB will go into the nucleus and try to bind, but it's being competed out, and unfortunately we don't have an upregulation of these 3 things, and cells that express ApoE4 will have a blunted autophagy response. So in summary, AD33 have better retrograde transport and less p-tau than AD. AD33 has a greater, more robust autophagy response compared to 4,4 based on protein and mRNA levels. AD44 and 3,3 have similar amounts of nuclear TFEB, so in other words, that system to upregulate nuclear TFEB is working fine, but they don't reflect that in their mRNA. And this is because ApoE4 competes with TFEB for the clear sites, interfering with the transcription of autophagy proteins. And this could be a reason why ApoE4 increases risk for Alzheimer's disease. So I wanted to leave you with this metaphor. So if we think of autophagy as eating the cells, this is an age-matched control non-demented patient right here. And this is what happens in AD. Okay, I just wanted to thank everybody who worked with me in the lab, especially Sundram right here, Dr. Barger's there, and Dr. Reese, all of our committee members, Pam and Dr. Griffin, of course, my good friend Rachel. Our whole lab group is depicted right here in this really nice picture. All of our techs and postdocs and that kind of thing. I wanted to acknowledge these people in Dr. Griffin's group in particular. Joanne has helped me a lot. Richard, who just recently retired, has helped me a lot. Dr. Ling Lu, and Orwa Abood, who's already graduated. Again, thanking all of our committee members, including Dr. Reese, Dr. Barger, Dr. Crooks, and Dr. Nixon, who could not be here with us today. Again, my friend Rachel Sundram, the geriatrics department for helping me out with all these other things, the MD PhD program for supporting this research, the Wingate family, the Johnson family, the Oppenheimer brothers, and the Riggs Benevolent Foundations, the Stop Alzheimer's Foundation, and of course the NIH grant AG1244. Are there any questions?
Congratulations, that was very wonderful.
Thank you very much.
And I'm so glad all of you came. It was great. And of course, I want to recognize this director of the PhD program here. And has been a constant support for us, always there. And we talk, or you need to talk much about Paulo. He's pretty good. And I also wanted to say just a few things before you have questions. And because I think that think that's reflected now as he goes from here to his program in psychiatry here at UMS, so he'll be with us and still working as an MD PhD. So I want to allow anybody to ask whatever questions they want. And although I thought when I was making so few remarks when I started, and we started a few minutes late, that I didn't want to say too much to take up time. But now we have plenty of time for questions because I read all the speakers and you finished early enough for your questions. So please.
Dr. Ozdemir. Oh. Rachel.
Paul, this was such an excellent presentation.
Oh, thank you.
How do we prevent ApoE4 from getting into the nucleus? What strategies can we maybe use to try to prevent that from competing with TFEB?
There's a couple of things that we had mulled over in our mind. Not necessarily the translocation necessarily. I'm not sure how we would go about preventing that. But one of the things that we could do, there are some groups that have these drugs that they've developed, small molecule drugs, that interfere with certain aspects of the ApoE4 site. And one of the things that I kind of glossed over was those residues that were important for this DNA binding. Those same drugs actually target those residues in particular. So I feel like if we design them ourselves or if we use these small molecules that could interfere with those residues for DNA binding, we could get a rescue from the interference of ApoE4 with the DNA and go back to ApoE3-like. That's something that we haven't gotten to start yet, but we'd hope to start to do that soon. Dr. Ozdemir?
That was so impressive. I think it explains very much how ApoE4 is a risk factor for Alzheimer's disease. So as a clinician, I want to ask, because nowadays we can do a lot of genetic testing, like people even can go online or buy these genetic cells, and they sometimes find out they are ApoE4 positive. So
Right.
as far as like from clinically, is there anything that we can advise them, or is there anything lifestyle, medication that we can advise them to increase their risk?
Sure. I think that's a great question. And like I mentioned to Rachel, I think we're kind of in the early stages of developing drug-like interventions. But we should say that despite the fact that ApoE4 is a risk factor for Alzheimer's disease, again, it's not a cause. It's not guaranteed to have it. I think there are a lot of other lifestyle modifications that we could advise in the absence of drug treatment, let's say. We minimize head injury, try to minimize being overly sedentary, getting more exercise, eating right. All of those things are modifiable lifestyle causes that we could recommend to people, especially if they have a family history or if they find out that they're ApoE4 positive. For example, autophagy is activated by exercise. So one thing that you could hopefully intervene in this is, if let's say you have ApoE4, maybe if you exercise more, your autophagy is going to be upregulated and you can prevent Alzheimer's disease. I mean, we're not quite to the stage of drug intervention, but we do know that diet and exercise do help. Dr. Rees?
So, Paul, very nice job. I wanted to, as you would say, get into the weeds a little bit.
Sure.
And ask you a couple of detailed questions. In your PLA slide for TFDB and 1433,
Yes.
you saw a lot of signal in the cytoplasm.
Right.
It was not appearing in the nuclei when you switched to AD, where the signal went down in the cytoplasm. So does that mean, since you're doing a PLA only, did you also test TFEB to see if it's down in the nuclei? Exactly. It's not appearing. It's disappearing from the cytoplasm in both of your AD samples, but it's not appearing in the nuclei. So that could just be that 1433 is not associating with TFEB. But you'd have to demonstrate that by showing that TFEB still is in the nuclei.
Right, and I didn't show the images of that, but this actually came from the red bars here. That was the measurement of the TFEB in the nucleus. So these two are sort of complementary results. We did see more TFEB in the nucleus in both 3.3 and 4.4, and at the same time, the cytoplasmic-only TFEB in 14.3.3 were in the cytoplasm, and there's less of them in AD. So we took that to mean that TFEB is entering the nucleus and dissociating with 14.3.3 in both cases.
Exactly. So, and then ApoE, interestingly, you showed in an electropherogram plot that you had more in the nucleus than in the cytoplasm. That makes sense if it gives Stone an equilibrium if it has a binding site on the DNA in the nucleus. But how do those levels compare with the levels in the circulation? In other words, is this a minor role for ApoE, or is it actually maybe its main role?
Well, I should say that this data here actually came from a cell culture model, so it'd be hard to translate this, I think, into a living organism, say. And also, these cells are overexpressing ApoE, so they are expressing it to a greater degree than would be physiological in the normal. The question of whether or not there is ApoE in a living human or a living organism's neurons is still somewhat open. We haven't really been able to do it ourselves, although we're thinking to do it soon. We suspect that there would be a lot more ApoE in non-neuronal or extracellular places, but I personally haven't looked at it.
Well, could I just intervene for 1 second?
Oh, sure.
Of course, Srini is doing these kinds of experiments, and it's not exactly the same as what you're asking about, because you're asking about the nucleus versus the cytoplasm. But these kinds of studies can be looked at and, really examined to see whether or not, for example, is a 3-3 person compared to a 4-4 person, what about the 3-4 person? So these are the kinds of challenges that we already, Srini and all of us have already talked about in meetings before. But these are the ways that we can get at what Dr. Osborne just mentioned. How can we take this to patients to help us think about what can we do? And of course, one of the big things, of course, is from Rachel and Barbara's work on the risk factor that's conveyed by obesity and type 2 diabetes. So if you have a patient that comes to you and says, I found out from 23andMe that I'm 4-4, and you say, what can I do? Well, you can talk about it on exercise, for example. But these are the kinds of things that you need to, just like you warn them to get their shingle shot or any other thing, and when we know risk factors, and now we're kind of become aware of this risk factor, because we send 150 bucks for the training find out. And so, since this is the case in your patient, it's so good, and the answer is so good, and there are things that we can do to encourage. So taking care of that risk factor is in the future, or maybe tomorrow. Maybe somebody's working on it right now. But that's in the future. But the now is we know about these other risk factors that are good in the population.
I think, ma'am?
Thank you very much for a great presentation. I really enjoyed it. The part that particularly caught my attention is when we were talking about your starvation model.
Yes.
So you have a large response in your E3s, and it's a lot less in your E4s, but you still have a response there. So
Yes.
do you think there would be a dose response that, like, you could just push that pathway up farther, and then you would be able to bring those E4s at the same level?
Well, it would be, I think, a little bit difficult. Since this is not a drug treatment, all we did was a starvation experiment. We starved them, without amino acids.
Serum starvation?
Yes. Mm-hmm.
But then if you would use something else like metformin or resveratrol that also activates autophagy, would you be able to get to something like that?
That's a very interesting question, and I think I would love to try it, but unfortunately we haven't gotten the chance to do so. TFEB also upregulates itself, so if it turns out that maybe you're upregulating TFEB or you're using these drugs to upregulate TFEB, since it's its own promoter as well, it might be difficult to overcome. But we'll be more than happy to try it. Dr. McGee? Oh,
So I was going to follow up on the 3-4, but are there any polymorphisms in 4 that super activated or anything that's been described where you see a worsening of it?
as far as I know, the ApoE4 itself is all that's been characterized. Any further polymorphisms I'm not aware of personally.
So I hope that you got permission from Pepperidge Farm for copyright on the P62 autophagy slide.
Yes, right. The goldfish. Right?
And then I'd kind of like to take old school. So I've been reading a lot about some things related to deer hunting in Arkansas. Could you talk briefly about the intersection between prion disease and Alzheimer's?
Yes. So prion disease is a disorder where these proteins that have a specific shape can spread through the brain and through cells based on the conformation of that protein, causing other proteins of the same type to have that same conformation, and it can spread this way. Mad cow disease, for example, is one of those. There is some evidence that people have shown where they took the hyperphosphorylated tau that exists in Alzheimer's disease and put it into a mouse and saw it spread in a similar prion-like manner. I think there has been a lot more of that coming up. I'm not sure if I totally believe it myself, but there's been some evidence for that.
It's very controversial. Just because of CWD, I've been reading a lot about prion disease. It always comes back, at least especially in the last few years, to Alzheimer's disease.
Right. So it would be like a tauopathy. Basically, the other tauopathies are more serious. Like they've shown other conformations of the tauopathies, and they form this prion-like process as well. And that's why some people are developing these antibodies against tau to see, try to get rid of the tau. I'm not sure if I'm fully convinced, but there is a lot of evidence for it. Yes.
Well, that was a fantastic talk.
Thank you, Dr. Rees.
Well, just to clarify for anyone who might have misunderstood this, there is no evidence that I'm aware of that the prion protein itself is activated in Alzheimer's.
Right,
It's
right.
just that tau can spread by a conformational mechanism like a prion.
Yes.
And possibly the A beta as well. So, Paul, to steer you back towards therapeutics, there are activators of autophagy that unfortunately have adverse side effects, so they're not likely to be widely used.
Right.
But caloric restriction and intermittent starvation are regimes that are perfectly acceptable, have a lot of other fringe benefits in terms of overall health. Have they been tried systematically on AD patients at an early stage? And specifically, if they have, is there a difference between the way 3-3s and 4-4s and 3-4s, for that matter, respond?
Wow, that's an interesting experiment. I should try to do that. Unfortunately, I'm not aware of the literature of anyone having tried that. Autophagy experiment or starvation experiments and things like that for humans is notoriously difficult because people don't like this protocol for the most part. But I'd love to see that tried. So far, autophagy and ApoE4 are not really well related. Not very many papers that are published on it. There are some, but there are not very many. So I don't think, even if that has been tried, I don't think that they'd have separated them by ApoE genotype.
Well, they already have Alzheimer's disease, so now you're going to starve them.
Well, and they might have FTE.
In a mechanism, that is all about mechanism, and so it'd be very interesting to test the idea because there are that adds to the clinical ideas about.
We are still doing chemo.
Dr. Yadavar.
Nice talk, Paul.
Thank you.
I have a comment on, FOE4s actually have more protein aggregation, and you see more and more of E4 getting segregated into the aggregates compared to the E3 patients or E3. So you think there is an evolutionary perspective? I mean, if you can tie autophagy and how FOE4 can compete for a clear side, is it? Do you think there could be an evolutionary reason why? Maybe there's a method for the madness of increased aggregation in E4 individuals because
Right, that's a great question. I think there's two parts to it. So you were mentioning that the aggregates that have of cells that express ApoE4 will have more ApoE4 in the aggregates. So that question, there has been a lot of evidence of ApoE4 having more preferential binding to the amyloid beta protein, for example. So that could be one reason why you're getting more ApoE4 in aggregates, because of its association with amyloid beta. I think the other answer to your other question is about the evolutionary mechanism for why ApoE4 still exists in the human population despite being, bad. There is some very interesting studies that have been done that when you inhibit autophagy in an immune cell, for example, it will overexpress its neuroinflammatory cytokines, including TNF, IL-6, and all that kind of thing. So you could imagine a cell that has ApoE4, they would be more likely to respond to external attacks. In addition to that, cells that have ApoE4, or people with ApoE4 in their brain, will also express arginase 1 to a greater degree, and that would tend to lead to less arginine in the extracellular space. And this is actually protective, and you can imagine a situation where you have internal parasites or things like that. You would want a greater inflammatory response in that population, and you'd also want to deplete arginine, because arginine is an important amino acid that parasites will try to get from the cells. So that's at least one mechanism for why ApoE4 might persist in the population despite having some of these effects. And furthermore, ApoE4 that we're showing right here in terms of Alzheimer's disease, of course, is a very late stage thing that will show up. So it might have a sort of an antagonistic pleiotropy effect there, such that it's helpful when you're young, but when you're older, it doesn't really help you very much, and evolution doesn't really help for that.